We are live.
Yeah, okay.
Yeah, Aditya, you can. Ashutosh, right? Then you can start.
Yes, yes, I have a presentation that I will be screen sharing.
Hello, everyone. Welcome. The event will start in 2 minutes.
Good afternoon, one and all. We welcome you to the day two of Nobelium 2020. The first talk of the day is by Dr. Soma Sanyal on this year's Nobel Prize in the field of physics. Dr. received her PhD in 2004 working on particle physics and cosmology from the Indian Institute of Physics in Bhubaneswar. Later, she was a postdoctoral research associate at Indiana University, Bloomington, where she worked on the rheological aspects of forms, among other things. Presently, she is an assistant professor in the School of Physics at the University of Hyderabad. Her areas of research include the early universe, cosmological phase transitions, and relativistic heavy ion collisions. The Nobel Prize 2020 in Physics was divided. One half awarded to Roger Penrose for the discovery that black hole formation is a robust prediction of the general theory of relativity, and the other half to Reinhard Genzel and Andrea Ghez for the discovery of a supermassive compact object at the center of our galaxy. In this talk, we will discuss their work, which led to the 2020 Nobel Prize for Physics for the three scientists. Lastly, a reminder, if you have any queries, type it in the chat box so that we can address it at the end of the talk. Now, without any further ado, we share the screen to you, ma'am.
Uh, Good afternoon, everybody. Can you see my screen?
Yes, ma'am.
Yes, we can see, yeah.
Just 1 minute. I'll just switch on my video and then we can get started.
Oh,
Okay. So I would right at the beginning like to thank the Junior Science Club and the Entropy Club for giving me this opportunity to talk about the Nobel Prize in Physics for this year, 2020. And I will try to explain it in a way that is non-mathematical. Actually, black hole physics requires a lot of mathematics, which is not accessible to all of you, which you are probably not acquainted with. However, what I will try to do is try to break it down in a way so that it is accessible to all of you. However, if you have questions, please do ask me. And also at the end of the talk, I will give you the references. So if any of you are interested to probe all these things further, you can go ahead and probe them using those references. So today we will talk about the Nobel laureates of 2020. And as was just mentioned by the person introducing me, one half of the award went to Roger Penrose, who is a mathematician and a theoretical physicist, for the discovery that black hole formation is a robust prediction of the general theory of relativity. And Roger Penrose, as I said, being a mathematician, all the derivations that he did are quite difficult to present in a short 30-minute lecture. So as I said, I'll try to present what was the most important thing that came out of all the things for which he was awarded. The other half went jointly to Reinhard Genzel and Andrea Ghez for the discovery of a supermassive compact object at the center of our galaxy. Over there too, I will just try to explain what exactly got these two people the Nobel Prize. So we'll start with Roger Penrose. I'm sure many of you are already familiar with his name and the name of Stephen Hawking, who worked very closely with him. And I'm sure that if Stephen Hawking was present today, if he was alive today, he would have shared this Nobel Prize with Roger Penrose, because much of their work are combined. But we'll start with the first paper that rolled this whole idea of space-type singularities and black holes. And this was the PRL paper published in 1965. I've given you a screenshot of the paper, Gravitational Collapse and Space-Type Singularities. This was by Roger Penrose. And the paper is a milestone in the history of general theory of relativity. So let us see what was so important about this paper. We'll start with the general theory of relativity, which was presented by Einstein in 1950, where he said that the gravitational field gives shape to our spacetime. Einstein's field equation can be written down in compact tensor form that I have written down on the left-hand side of the screen. I will briefly tell you what these things mean so that you get a feel of what is so special about general theory of relativity. So over here you see some new characters, one of which is the G with the subscript of mu nu and the other T mu nu. If you already know tensors, then you know what is written over here. But even if you don't know tensors, I'm sure all of you are familiar with matrices, and I'll try to explain how G mu nu is important for understanding the space-time and how T mu nu is important for understanding the mass and energy of an object. So basically, Einstein's field equation gives you a relationship between this G mu nu and T mu nu. G mu nu is the space-time characteristic, while T mu nu talks about the mass of the object. The G mu nu, or the metric tensor, defines the coordinate system that you are working in. And once you have your coordinate system, you'll be able to find out the line element on the coordinate system. That line element will tell you whether it's a flat surface or a curved surface. So for example, I have shown two G mu nus in the two matrices. And you see that the two matrices are different because their diagonal elements are different. The first matrix on the upper right-hand corner is the one that represents our flat space-time. How do you reconcile it? Just think of a straight line and the equation of a straight line. You know that it is x square plus y square plus z square. Here we have another dimension to it, and that is the dimension of time. But you know, in physics, we always have to check and see that all the dimensions are correctly matched. And since t is a time dimension and x, y, z are all spatial dimensions, so you have to include the velocity of light c. That's why you see a minus c square dt square at the beginning of the right-hand side. That makes the fourth dimension also space-like, but it is actually the time. If you have done special relativity, you will be familiar with this already. This is what is called the Minkowski space, the 4 time and space dimension for flat space. Now look at the sphere. If I want to draw a line on the sphere, I generally use the spherical coordinates. And in the spherical coordinates, when I write down the metric is different. So therefore, when I'm describing the line element on the surface of the sphere, then I have used a different metric and I have used a different surface. So therefore, the nature of G mu nu will tell me whether I am on a flat surface or I am on a spherical surface. So therefore, G mu nu tells us whether space is curved or flat. What about T mu nu? T mu nu is what is known as the energy momentum tensor. It is different for different forces. Like for example, you can write down the T mu nu for electromagnetic force also. You can write it down for gravitational force also. It is nothing but the energy and the momentum tensor. Now in momentum, you already know that mass is there. Mass is a part of momentum. What about energy? Well, if you have done special relativity, you already know that energy is another component or the temporal component of the 4 momenta. Even if you have not done special relativity, I have put up the expression for energy on the right hand side, and you can see that it is E square equal to P square C square plus M0 square C to the power 4, where M0 is the rest mass of the particle. So this tensor is mostly dictated by the mass of the object that you are looking at. So on the left hand side, we had the G mu nu, which was the space-time characteristic. On the right hand side, we had the T mu nu, which is dictated by the mass of the object. In a nutshell, Professor John Wheeler had told us that general theory of relativity can be summed up in this sentence. Space-time tells matter how to move. Matter tells space-time how to curve. This is a very famous statement by Professor John Wheeler. And as you see, if you look at the small red ball, it rolls down the hill because that is how the space or the manifold is made. So it will roll down the hill due to gravity. Whereas in the case of the huge mass of the Earth, it is changing your space-time fabric itself. The checked thing that you see is the space-time fabric, and it is making a small dent in the space-time fabric just because of its sheer mass. Now let us look at singularities in spacetime. You have probably heard of singularities in some mathematical equations. You have got some terms where you have 1 by x, and you know as x tends to infinity, as x tends to 0, that particular term will become undefined, or you will get a singularity for that particular term. Now, what is a space-time singularity? A space-time singularity is a breakdown in space-time. It can be in geometry or it can be in some physical structure also. Unfortunately, what we find is that Einstein's equation has a space-time singularity. The space-time curvature values as well as the matter energy densities start to diverge. That is, they become very, very large, just as your 1 by x term as x tends to 0. Einstein himself was very uncomfortable with these singularities in the general theory of relativity, but he could not really do anything about them. They were there in the theory that he had presented, and he didn't have any way to explain them physically. The era after Einstein. Einstein died in 1955, and the same year marked the publication of the first singularity theorem by Amol K. Raychaudhuri. If you have been following the newspapers, following this year's Nobel Prize, you may have seen that many people have written about the India Connect to this year's Nobel Prize. The India Connect is nothing but the Raychaudhuri equations put down first by Amol K. Raychaudhuri, and it is a very notable milestone in the history of general theory of relativity. Professor Raychaudhuri received his PhD from the University of Calcutta, and one of his thesis examiners was Professor John Wheeler, whose quote I had just told you about. And this Raychaudhuri equation later on led to Penrose framing his new theorem of singularities. So that is the connect of this year's Nobel Prize with India. What did A.K. Raychaudhuri do? Well, A.K. Raychaudhuri used the concept of geodesics to understand singularities in space-time. The question arises, what is a geodesic? A geodesic is a locally length minimizing curve. It means if you have 2 points, suppose 2 points in a room, and you want to move from one point to the other, what is the shortest path that you can take? Obviously, the room being a flat space, it is a straight line. However, it will not be the same for other surfaces. Suppose you are on the surface of a sphere, that is how our airplanes fly, then the geodesics are no longer straight lines, but great circles. So all the airplanes try to go on the great circles when they're flying a long distance between two different continents. Thus, a geodesic is different from surface to surface. The geodesic also tells you how matter flows along the surface, that is the manifold, when there is no force accelerating it. Whenever you have a singularity, it automatically means that some physical property of this fluid becomes unbounded. In usual cases, it indicates that the density of the fluid becomes unbounded. And these geodesics, which do not end up anywhere, are called incomplete geodesics. So, what has all this to do with Penrose's contribution? Well, what many people started saying is that maybe it is because of the spherical symmetry that we have and that we use, we are getting these singularities in the Einstein's equation. That is because if you have a 1 by r and there is a r equal to 0 point, you are bound to get a singularity. However, Penrose wanted to prove that the deviations for spherical symmetry would not prevent the formation of singularities. So it is not because there is spherical symmetry in the metric that we are getting these singularities. Whatever metric we take, we will always get these singularities. So, wanting to prove this, Penrose brought in the idea of incompleteness to describe singular spacetimes, and he introduced a new notion of closed trapped surface for the first time. This was the concept that was introduced in the PRL paper that I had showed you. And this was a major conceptual contribution to the physics of gravitational field because it helped you to physically visualize what happens when you encounter a singularity. What he showed that there are closed space-like surfaces such that their area tends to decrease locally along any possible future direction. And he showed them with the help of the geodesics that had already been used by Amol Kumar Raychaudhuri. What about black holes? Now you know that the conventional definition of black holes is that of a region from which nothing comes out, not even light. Now the pictures that I'm showing you, please remember, they are just artists' rendition. They are not real actual pictures of any black hole. So please do not take them very literally. They are from the NASA web pages, and they are just artists' rendition of how a black hole might look like, but they are not real pictures. So please do not consider them as real pictures. Now, gravity inside a black hole is so strong that matter is pulled into the hole, but it cannot escape. You also see a line in the picture, which is supposed to be where the singularity is. But again, please remember, this is a artist's rendition only. This is a black hole which is accreting due to mass from a star. The star is the white one on the right-hand side. So all the mass from the star is entering the black hole, but nothing is coming out. That's what a black hole, or how a black hole is defined. What have black holes got to do with these trapped surfaces that Penrose wrote about? Well, a singularity will indicate a break in the geodesic. Geodesic, for in this case, means a continuous chain of events through space-time. So it can be something like I am now speaking. So I am following a geodesic in time. I am stationary, so I am only following a geodesic in time. So it's a chain of events which is occurring through space and time. Now, if you have a closed trap surface, which is a two-dimensional embedded submanifold, it is compact without boundary. Now, you don't have to bother much about these particular words. All you have to understand is that it is such that you cannot leave the two-dimensional surface at all. Once you are on the two-dimensional surface, you are stuck to the two-dimensional surface. So therefore, a ray of light on the two-dimensional surface will form a circle. That is what I have tried to sketch on the right-hand lower corner. You see that rectangle is suppose your manifold or the closed trapped surface, and on that the red and the orange are light rays which are trapped forever on the circle, and they cannot go out of the circle. So you have given a physical understanding of what it means that the light enters but can never leave the black hole. So light rays cannot escape from closed trapped surfaces. This was what had been bothering everybody very much, because in a singularity, how do you explain it physically? And this was given by Penrose. Of course, this was not complete at that time. After his first paper, Penrose and many other people started working on the singularity theorems, and Professor Wheeler was also one of them, and their understanding of black holes. Finally, Penrose and Hawking came up with another theorem that established the concepts of space-time singularity and was instrumental in formulating the proper theory of black holes as we know it now. It also established that there was no closed time-like curves, and that the concept of trapped surface is local, that it is not something that it's trapped, which will have something in the future or the past. So the timeline is not compromised. It's only in the space. The trapped surface exists only in the space. So that was the contribution of Penrose in the understanding of the black holes, for which he got this Nobel Prize. We'll now move on to detection of black holes. As I've just mentioned, if no light comes out of a black hole, how do we know that it is there? Well, even though we cannot see the radiation from the black hole, we know the presence of black holes due to the motion of matter around them. As you know, the black hole has a very strong gravitational field. And this field affects the stars and the gas which are moving around the black hole. Also, it has been seen that when a black hole and a star are close together, there is a high energy radiation that is emitted. Though this radiation is not in the visible range, that is, we cannot see it with our naked eyes, this can be detected by telescopes. So, while people have predicted black holes, they have also predicted ways to detect them. And astrophysicists and astronomers have been trying to detect these black holes for a very long time. There are also what are called supermassive black holes. And these black holes have masses which are more than 1 million suns together. Really very heavy black holes. And scientists have also found proof that every large galaxy contains a supermassive black hole at its center. The supermassive black hole at the center of the Milky Way galaxy is called Sagittarius A, and astronomers have been tracking this and trying to map this for a very long time. It has a mass equal to about 4 million suns and would fit inside a very large ball that could have take 1,000,000 Earths. How did they map all these out? Well, that is where the contribution of Andrea Ghez comes in. And astrophysicists, they have been mapping using both land-based as well as satellite-based telescopes. You all must have heard about the Hubble telescope. That is a satellite-based telescope. But the galaxy, our galaxy, is typically mapped with ground-based or land-based telescope. And these are the picture of the two telescopes, the Keck telescopes in Mauna Kea in Hawaii, the island of Hawaii. Mauna Kea is a volcano. So these are administered by the laboratories in some of the American universities. And Andrea Ghez, who is from UCLA, worked all her life on these telescopes. What she was trying to do was develop techniques for high angular resolution of the astronomical imaging. And she also perfected the use of adaptive optics and tried to get more and more details of the supermassive black hole that is at the center of our galaxy. She has been working on this for the last 25 years. And in 2019, they had a science paper which talks about the galactic center supermassive black hole and the relativistic redshift of the star, SO2. This paper conclusively proved that there is a supermassive black hole at the center of our galaxy. Hers, of course, was not the first work. Above that, we see the paper by Reinhard Genzel, which was published in 1996. It's a Nature paper, and there they have clearly mentioned that there is evidence for a massive black hole at the center of our galaxy. Both these observations together have now established beyond doubt that there is a supermassive black hole at the center of our galaxy. Of course, it took a lot of time and a lot of mapping. What they had done is they had gone to lower and lower length scales. This is like once you want to look at Google Earth pictures and you want to see our university, you first see India, then you go down and find out where is Hyderabad, and then finally you find out where the university is, and you're happy to see that you can see your hostel buildings. So there is absolute proof that you can see it from these images. Similarly, Andrea Ghez and Reinhard Genzel both worked very hard to get down to very lower length scales and do the mapping to establish without any doubt that there is a supermassive black hole in the center of our galaxy. I will stop here and I will give you the references. As I mentioned, it is very difficult to bring out the nature of this work in a short talk like this. However, what I have tried to give to you is a picture of the theoretical and the experimental nature of this work. And the thing is that at the end of the day, the theory and the experiment came together in a beautiful way. And that is why this year's Nobel Prize is kind of a celebration of the unity which we see in physics of both theory and experiment. And that is what makes me very happy. These are the references that I have used for this talk. A lot of these pictures were taken from the NASA website. And there are a few papers which I have also mentioned. As I said, if some of you are interested in more mathematical aspects or understanding these things a little more deeply, you can, of course, let me know and I will help you. So I would like to take any questions that you have.
Ma'am, we firstly would like to thank you for that wonderful and insightful talk about black holes. Now, we were doing a live streaming on YouTube and we seem to have a couple of questions. So, maybe go ahead with that.
Yes, sure. Please go ahead with that.
So, the first question we had was, could we have obtained as much knowledge about black holes and their formation if it wasn't for the general theory of relativity?
Uh, well, the mathematical basis of the black hole is the general theory of relativity. So if we did not understand the general theory of relativity, we won't be able to understand the reason why there is a black hole or what a black hole means or how we can go around detecting a black hole. So yes, we need to know general theory of relativity if we really need to understand black holes.
Okay, ma'am. The second question which came up was, how much has the discovery of gravitational waves enabled Dr. Penrose's work to be proven right?
Well, as I just tried to explain, it's not really gravitational waves that Dr. Penrose worked on. The theorem that he has been awarded for is basically on collapsing stars. It's not really on gravitational waves and the formation of black holes, the singularities. The singularities are very difficult to understand physically. So mathematically, people used to get this. Even Einstein, as I said, was very uncomfortable with the singularities in his theory because he couldn't really understand them physically. So that was the discomfort that people had. Dr. Penrose's work basically gave a physical insight into these singularities. It kind of connected the black holes to the singularities. So that is why Dr. Penrose's work was given this Nobel. It's not really to nothing, and it doesn't have anything to do with gravitational waves as such.
Okay, ma'am. Thank you. And one more question we had was that, have there been any other individuals of an Indian origin who have contributed to the general theory of relativity?
Well, there are plenty of people who have contributed to general theory of relativity, but the reason why I mentioned A.K. Roy Choudhury's name because he's probably one of the most famous. I still remember a conference that I had gone to, and while we were standing in the queue for tea, I remember at that time the Royal Astronomer of England, he was there. He was the chief guest for that evening's talk, and he came up to A.K. Roy Choudhury, who was standing there in front of me and said that, sir, I have heard about the Raychaudhuri equations all my life. I'm pleased to meet the person himself. So that was his stature. That was his stature in the scientific world of general theory of relativity. That is why I thought that it would be a good thing to at least mention him in this talk. But there are
Thank
many.
you. Thank you, ma'am, for that wonderful anecdote. But one of the participants, Deepika Parthanasarthi, asked that I've been quite interested. Can Dr. Sanyal recommend any material that could get me started on this black hole formation?
Yes, of course. I don't know your background, which year you are, but maybe what you should probably do is start on with tensors and all these things, and then get a book on general theory of relativity. That is the first step, but you have to do the mathematical background first. So that will require you to learn a bit of classical mechanics and all these kinds of thing. Why don't you just send me a mail at my email address? That would, I can give you some more pointers then directly.
Okay,
Is that
ma'am. We'll share your email ID if possible with the participant.
sure? Sure.
One more question which came up was, could Penrose's discovery tell anything about the origin of our universe?
Well, the origin of our universe is also believed to be through a singularity. And therefore, of course, again, as I said, people are uncomfortable with singularities. So, yes, Penrose, in a way, gave us a way to look at singularities where we can have a physical understanding of it. So, in that sense, yes, it can help us to understand the Big Bang. But as of now, there is no exact theory of that. I mean, there are many, but we don't know one which is as well established. In this case, it is now very well established by the experimentalists.
Okay, thank you, ma'am. And the next question we had was, does the movement of other astronomical bodies get affected to the high gravitational pull of such supermassive black holes?
Yes, they do. The high gravitational pull of these massive black holes does affect the other bodies, astronomical bodies, which are coming into its periphery also. If, as you know, that whenever we are close to a gravitational field, for example, we are here on Earth, right? And Earth has a gravitational field. So when we jump up, we come down. So there is a jump up and down due to the Earth. Suppose I generate a velocity which is greater than the Earth's escape velocity. Then when I jump up, I'll just leave the Earth and go out. So it depends on what is the mass and momentum when you are doing interactions. Similarly, if some massive object comes close by to a black hole, then the black hole will, of course, affect its motion. And it can even start accreting matter from that.
Okay, thank you, ma'am. And the last question from the participants will be, is there more than one way to detect black holes?
All the different ways are usually through the motion of the objects around the black hole. So what people try to do is try to map these luminous objects, because what we see through the telescopes is mostly the radiation at different wavelengths. Luminous does not necessarily mean only for visible light. It can be in the other ranges also. So what we are trying to do is mapping these things. And mapping these things is the only way that we can detect black holes. There is, of course, other radiation. However, we are not really sure whether this is emitted by black holes or not. The best way till now is to map the massive objects moving around the black hole.
Thank you for that, ma'am. Now, the faculties and the other distinguished professors present in this meet have some questions. So we request them to unmute themselves and ask.
Can I just answer a few more questions to supplement Professor Sanyal?
Yes, sure. Please go
Yeah.
ahead, Bindu.
Well, first of all, there are many Indians who have contributed to the theory of general relativity and black holes, including S. Chandrasekhar, who actually proved before Rai Chaudhary, he proved that there was a limit at which stars collapse, called the Chandrasekhar limit, and then that led later to the limit of the singularity limit of the black hole. Then there also have been Vaidya, P.C. Vaidya, who was one of the first people to find a solution to Einstein's equation, which is also very much used in black hole physics. And then there is Professor Narlikar, Jayant Narlikar, who proposed an alternative theory to the Big Bang theory of the universe with Fred Hoyle. And but that theory later was proven wrong, but it gave rise to a number of interesting speculations as to what happened before the Big Bang, and which Penrose is now working on, and it's called something called the bouncing universe. What happened before? How the Big Bang came about is what Penrose is now working on. So there have been many Indians, and then there is somebody called Abhay Ashtekar who worked on other ideas of Penrose about how space-time is generated. Penrose has done a lot of work in many areas, and has had many Indian collaborators. The best of whose is Abhay Ashtekar, who worked on some of those ideas, and those are relevant to getting a quantum theory of gravity now. And so that's one thing. And the other thing I want to say is that there are other types of black holes in the universe, and they typically occur in binary star systems. And one of them is called Cygnus X1. And what happens in a binary star system, one is a visible star and one is a black hole in the binary. So the visible star is the one whose trajectory you see. And from the trajectory of the visible star, you can conclude that there is a black hole, which is its partner in the binary system. So these are some of the supplements to the answers given by Professor Sanyal.
Thank you. Thank you, Bindu.
Ma'am, I have a small question.
Sure, go ahead.
Ma'am, you just mentioned Cygnus X1, right? Isn't that the first, if not one of the first black holes discovered? I just wanted to know. I was curious.
Yes, it is,
Yes.
but it's not in the center of any
Galaxy.
galaxy.
That's right.
Okay,
It is
thank you,
one of
ma'am.
the black holes.
Okay, thank you, ma'am.
Yeah.
Ma'am, we also had one more question from Sharmista ma'am. If you
Sure.
could unmute yourself and ask.
Hi, Soma. Really enjoyed listening to you. Being biologist can't help because for some time, last decade or so, the concept of geodesics have been into biological systems as well. Very universal.
Yes.
Is that okay to say?
Right. Because
Yeah.
you see over here also, it's always the same things: energy and momentum conservation, and that is what is there in, I think, believe in all parts of science. Energy. We
Yes,
always
that's
try to
fairly
conserve energy.
constant. Yeah.
Yeah. So it's basically the concept comes from energy. So
Yeah,
Yes.
that's
that will be the most stable configuration of a biological system. If on a
right.
given surface, it is
Right.
on a geodesic.
Yes. Thank you so much. Enjoyed listening.
Thank you so much.
With that, we will come to the end of this session for the 2020 Nobel Prize in Physics. We would first like to thank Soma ma'am for giving this wonderful session. We would also like to thank all the participants and all the faculties for giving their comments and questions and making this discussion more fruitful. Now, I sign off with the hope that everyone thoroughly enjoyed this wonderful session. I would like to thank Dr. Soma ma'am once more. With that, we have come to the end of this session. I request all participants to stay tuned as the second talk of the day will begin shortly.
Thank you, Gautam. Hello,
Yeah.
everyone. I think we'll be starting the talk very soon. Hello, Chandrasekhar sir. Am I audible to you?
Yeah, you are audible to me.
Yeah, hello, sir. Good evening. So, yeah, I think, shall we start?
Yeah, please.
Yeah, sure. Okay. Good evening, everyone. I welcome you all on behalf of Junior Science Club and Entropy to Nobelium 2020, which is a webinar series on all the Nobel Prizes given this year. This talk is on chemistry's Nobel Prize winning gene editing method, CRISPR, which has transformed the molecular life sciences. CRISPR, which was developed by Emmanuel Charpentier and Jennifer A. Doudna, enables scientists to precisely alter an organism's DNA, promising improved crops, new treatments for diseases, and more. Now, without further ado, and with extreme honor and pride, I would like to introduce you all to the speaker of our talk, Dr. P. Chandrasekhar. Dr. Chandrasekhar has obtained his PhD from the Center for Cellular and Molecular Biology, Hyderabad, where he worked in the area of stem cell biology and mouse genetics. During his postdoctoral research at Mount Sinai Medical School, New York, in stem biology at the University of Edinburgh, he focused on understanding the autoregulation of nanog and studying novel pluripotency factors pathways in murine embryonic stem cells. He was a Wellcome Trust DBT India Alliance Early Career Fellow at INSEM Bangalore from 2011 to 2013. Later, Sir joined CCMB as a senior scientist in 2014. Currently, Dr. Chandrasekhar is working as principal scientist at CCMB Hyderabad. His lab extensively uses genome editing stem cells and mouse embryos to study cell fate decision in pluripotent state and self-organization of stem cells in embryo-like structures. Now, without taking much of your time, I would request Dr. Chandrasekhar to start the session. A humble request to everyone in the audience is to type their question in the chat box and they will be addressed by Dr. Chandrasekhar at the end of our talk. Over to you, sir.
Thank you very much. I hope the screen is visible to all.
Yes, sir, it is.
Okay. Thank you very much for the nice introduction and giving me this opportunity to speak before you on the CRISPRs. And one correction to make is I'm not a CRISPR biologist. I am just one of the users of this great tool. As was introduced, I am just a mouse genetist who work on stem cells and the early development of mouse. So coming to the Nobel Prize of this year in chemistry, it's interesting that it was given in chemistry, not in medicine, but nonetheless, it's a great finding that has transformed the way we do biology these days. And this first slide is something I picked up directly from the Nobel website, where they call them as genetics is a tool for rewriting the code of life. Indeed, it's true that we can rewrite the code of life in almost all the organisms possible. And I think this is one of the only Nobel Prizes where two women were awarded jointly for the first time. So that makes it all the more special. So although Emmanuel Charpentier and Jennifer Doudna are the Nobel awardees of this year's chemistry award, but the journey of CRISPR starts quite long back in 1987 and was contributed by many people who built it bit by bit, and it culminated in this great finding where they developed these tools to manipulate the genome. So I will take you back to those days of 1987 when the first findings came out. So before that, what was our life like before having these CRISPRs? So the genetic manipulation is a major tool that's used by all biologists to understand the gene functions as well as understanding the biological processes. But getting them working in a precise, a targeted manner is very difficult. And such tools were available only in certain small lower organisms like the bacteria and in yeast, where the homologous recombination is having a very high frequency, whereas this is off limits to most of the advanced or eukaryotic cells, particularly the mammalian and the vertebrate cells, where the homologous recombination is very poor. And in case of mammals, only in mouse, there was a possibility of precise genetic manipulation because there were embryonic stem cells which had a higher frequency of homologous recombination and they were germline competent. So this was the only mammal that was amenable for any genetic manipulations. And also in whichever species it was working, be it bacteria or the yeast or other lower organisms, the process of getting a target specific genetic modification was very elaborate, time consuming and very expensive. Many a times in the cases where it works, you have to screen thousands of cells to get the desired event. In the end of 2010, 8 to 10, the other technologies came in. Zinc finger started way back in 90s, but they picked up the steam only after 2005. And other editing tools like Talents came up around 2010. They were workable. Zinc fingers were more difficult, whereas Talents were much easier. They were workable, but still they were difficult to adopt by different labs, as well as to set them up. And the ease of use was not convenient, so they did not spread across the globe, nor many people were able to use them effectively. So this is where we were, and in 2012, the CRISPR tools came and they have changed the way we do biology now. Now, going back to 1987, the first paper that had any hints of CRISPR, then it was not known as CRISPR, is shown here on the left. That's by a Japanese group, Yoshizumi Ishino, where when they were trying to study the sequence of a gene called IAP gene, they were sequencing. Those days, the sequencing was much different, as we all know. That's using the gels. So they sequenced this gene plus the sequences around it, and one beside the other, and that's where they named it for the first time as CRISPRs. Of course, the full form is really a troublesome one: clustered, regularly interspaced, short palindromic repeats, which describes basically there were these repeats and they were also associated with the Cas proteins. So at this time, they could only say that these were found in almost all the bacteria and the archaea, and this is the structure of the Cas locus or the CRISPR locus, but what is its function was not known. Then came 3 papers back to back in 2005, which are put here on the right. All of them, 3 of them independently showed that these repeat sequences that they were seeing had resemblance to the foreign genetic elements or the bacteriophages, suggesting that these were the source of these repeats was for the first time discovered. And later on, people went on to analyze them. And if you see here in this slide on the right, the Biology Direct paper was the first one from Eugene Koonin's lab, which suggested for the first time that these systems had a similarity to the RNA interference, because RNA interference was pretty well known by that time. They suggested that this has similarity to the RNA interference system of the immune system that is known in the prokaryotes, and probably this is having a similar sort of an RNase and a RNA, which lead to an acquired immune response towards this the bacteriophages or the foreign genetic elements. So this was a paper that suggested it, and soon in 2007, a science paper came which conclusively established that, where they showed that they infected the bacteria with a virus, and then they showed that that DNA was taken in and it gave them an acquired resistance against the virus in the later stages. The surviving bacteria were resistant to the virus. So this one established for the first time that this is an acquired immunity against the viruses. Then a lot of groups got interested in it, and one of the group was Emmanuel Charpentier's. Her group also started working on it, and it took them a couple of years before they came up with this paper that is in Nature, where they showed that the CRISPR system, what and all were there and how they were functioning, but the exact functioning was not known. What they showed is, if you see here in the bottom, there is a CRISPR locus which is shown in the rhomboid green. The green rhombus, what you see are the repeat elements, and this is the CRISPR RNA or the cRNA. And in this paper, they showed that this CRISPR RNA, for maturation and loading onto their function or whatever the function they have to do, they needed to be matured. And this maturation was possible by another set of RNA which was adjacent to it, that is called the tracer RNA. This tracer RNA bound to the CRISPR RNA, and now it was serving as a target for the RNase3s, that were RNase3 class of enzymes that are there in the host itself, which will process this and make it mature RNA. So the processing for the first time was described in this paper. This was the first insight into the sort of mechanism. At this time point, Emmanuel Charpentier met Jennifer Doudna, and they both collaborated. Jennifer Doudna, as we all know, is a structural biologist. They both collaborated to understand exactly how the Cas proteins here utilize the cRNA and the tracer RNA to target the genes or the DNA sequences or the target DNA sequences. And that's what led to this paper, what we know, one of the famous papers, which gave them the Nobel Prize. That's a programmable dual RNA guided DNA endonuclease in adaptive bacterial humanity. So this paper is interesting because this is the landmark, and this is the reason they got the Nobel, because this describes the complete mechanism of how the CRISPR works. So if we look at this in the cartoon here, the phase 1 immunization acquisition, this part was already well established, as I said. Whenever there is a viral DNA that is in fact infecting the bacteria, the Cas proteins take a small part of this viral DNA and then they stitch it into the CRISPR array to keep it as a memory, so that this serves as a library. And in future, if the bacteria infects them, that can be taken care. The bacteria is infected by the virus, it can be taken care. Now, what this paper showed is the phase 2, or the defense resistance, how it exactly works. So what they showed is it also has the tracer RNA beside it, which is continuously transcribed. Then you have the CRISPR locus that is continuously transcribed. Both of them are together processed by the RNase3 to create the guide RNA. So they, for the first time, showed the CRISPR system here contains two RNAs. One is a tracer RNA and the other is a cRNA, or the RNA that is specific to the sequence. Together are loaded onto the Cas9 protein. Then the Cas9 protein is able to target the target DNA, and it creates a double strand break. And they also clearly explain how the double strand break is created, which I'll just mention in the next slide. And by this, the invading RNA will be degraded. So bulk of these experiments were shown in the test tube, and these are clear-cut molecular biology experiments which showed that, or conclusively established, this is the mechanism. So there are three components. One is an enzyme and two are the RNA. At the same time, this paper got published in August. Another lab, that is Virginius Sickness, this group also had independently found out the same mechanism. In fact, they sent it to Cell, which I think because of the review process and the delay, they got rejected. Then later on, it got published in PNAS just 3 months after the Charpentier and Doudna's paper got in Science. So yes, the priority of the discovery is with these two ladies, but we should not also forget that there are others who have been contributing to it, and Virginius Sickness is one of those who independently had the same finding. And of course, it was later on recognized by Kavli Prize, which was given to actually 3 of them. Okay, now getting into the details of that paper, what they clearly showed is the mechanism how the double strand break was created. So they described that there is a protospacer adjacent motif, which in this case, which is a Cas9, is NGG. This is where this happens as a point of stop and scan for the Cas9, where once the Cas9 is loaded with the guide RNA, it scans the DNA and locks onto the PAM. And from there, it tries to open up the DNA and see if there is a homology. And if the homology is not there, it fails and leaves, and it keeps scanning. So once there is a proper homology available, then there are 2 nucleus domains in this Cas9. One is the HNH and the other is the RUVC. which each one of them will cut one strand of the DNA, creating the double strand just 3 bases before the PAM. And this is a way double strand could be created. Later on, people have shown that you can either mutate one of them and create a single nick, or you can mute both of them, and it's called a dead Cas9, which can go and scan and bind to the DNA, but is not able to create double strand break. So the mechanism was completely described in this. Now, all these experiments, as I said, were done in the test tubes. And then comes, this is a paper which most of us relate to and remember, that came in February in Science by Feng Zhang's group, because this is for the first time they demonstrated, which came after around 6 months, where they demonstrated that in vivo, within the cells, in the mammalian cells, this system can be used to create a double strand break. Now, what happens in the cell after the double strand break is described here. So once a double strand break is created, it has 2 paths to take up for repair in any typical cell system. One is non-homology enjoining, and the other is the homology directed repair. Non-homology enjoining is a process by which the DNAs are sealed, but they do not have any reference sequence to see whether the DNAs were sealed as per what they were before. So there is always a possibility of losing some sequence or gaining some sequence, which leads to either insertion or deletion of the sequences, leading to what we call as indels, changing the sequence of the DNA. Whereas homology directed repair always has a donor or a reference sequence which is stitched into by homologous recombination, and you have a faithful repair of the DNA. So this is a very rare event. So the point here is the CRISPRs only create a double strand break in a cell. What happens after that is left to the cell. What CRISPRs do is to provide an exact precision tool which, based on the sequence of the guide RNA, will be able to locate to the specific locus. Unlike the restriction enzymes, which can cut in multiple places or any other nucleases, they can latch on to a specific sequence and create a double strand break. What happens after that is all dependent on the cellular conditions. So the results or the outcomes of the events are dependent on what is the cellular condition, what was provided, what was not provided. So later on, another CRISPR system was discovered, that is CPF1. In fact, Jennifer Doudna's group has also solved its structure. What they show is that this is one system where you don't need 2 RNAs. Instead, it has only single RNA. So the cRNA itself is sufficient, and the CPF1 is able to take care of the function of the tracer RNA. So just moving on, some of the utilities of the...
So there's a small request. So can you hide the pop-up at the down of your screen?
Pop-up. Okay.
Yeah, thanks.
Is it fine now?
Yes, sir, perfect.
Okay, I can't see the slides. Okay, fine. So I'm just going to put some of the utilities of CRISPRs, but as you know, there are many more. I'm just trying to restrict myself to some. The earliest ones that were to come were the transcriptional activation or repression systems, which use basically the dead Cas9. And for transcriptional activation, you can tag it or fuse it with a VP16 transactivation domain, which can recruit the activation complexes and the pol to the promoters and activate a gene which was silent. Similarly, a gene can be shut down, or its transcription can be shut down by using CRISPR interference, either by targeting the Cas9 to the just downstream of the promoter, or by using a crowd domain fused to the Cas9, which will bring in the repressor complex and repress it. And since there were indications of off targets in the beginning, of course, now there are CRISPR systems which have very minimal off targets. So photoactivatable CRISPRs were also created. Then you can do, since using this dead Cas9, because they can only locate to a locus but cannot create a double strand break, one can do all sorts of modifications into the DNA or the neighboring proteins that are there. So some of the examples are the epigenetic or genetic modulations, adding a methylation or removing a methylation or acetylation, and all these things can be possible and have been clearly demonstrated. Then the next generation of the CRISPRs that came are the base editors, here to create a change in the DNA without creating a break, because the break is fine for us, for biologists who use it in the lab. But when we talk about therapies, because this has a huge potential in therapy, people were trying to generate systems where can you create a point mutation or few point mutations without creating a double strand break, because double strand break many a times will have a undesired effect. So then came the base editors where you have a dead Cas9 tied to either cytidine deaminase. I have given an example here of cytidine deaminase. You can also have adenine deaminase, and you will be able to change the bases from either C to T, or the other one is A to C. Okay, and also you have RNA editors where instead of changing in the genome, you can have a transient change in the mRNA itself without making any genetic modifications. And Cas13b was the one of the Cas proteins, and there are multiples now known for editing the RNA. So the editing can be done in the RNA, and we know that the RNA editing is a very important regulatory mechanism, and we still don't understand a lot of them, but many of the modifications can be done using the RNA editors as well. So the impact of CRISPRs on biology has been immense. I think that's one of the reasons why normally if you see the Nobel Prizes are given, most of them, if you average them out like 15, 20 years after the discovery, there have been very few instances where the Nobel Prize is awarded within a decade of the discovery. In this case, it is just 8 years. And the other one I remember is the IPSCs, where it was given in a short time. These are the only few examples I know that which have got a huge impact within a short time. That talks about the implication this has on the field. So the genome editing now is demonstrated to be working in almost all sorts of the animals that have been tried. Previously, as I said, it was only possible in some lower organisms, and in mammals it was just in mouse, and it was almost impractical to do it in any somatic cells because the homologous recombination is very poor, so you can't do a directed change in the DNA. Whereas now it's possible to do it in almost all cell types and all organisms. So I think the spread of or the use of this in all sorts of organisms has made it a great tool. It's not limited to any single field or a single research area. And the best part about CRISPRs, why it became so popular, was it was an open access system. Once these things were developed, they were available on AdGene for anybody. Everybody could get it and could start using them. Now, that alone did not make it a favorite tool. The other greatest advantage of CRISPR is the ease of design. It's so easy to design. Why it fares well better than the zinc finger nucleases or talents is, in case of both zinc finger nucleases and talents, it's the protein that was recognizing a DNA sequence to target it. So there, for every DNA sequence, you have to design a new protein. In this case, the nucleus was same. It's just the RNA sequence you need to just change. 20 nucleotides you change, and you will be able to target a different sequence. So, and this is very easy to design because all the tools were available and online. People could give the gene sequence and would pick up guide RNAs. All the algorithms were worked out by multiple groups. And one can order the oligos and just clone. So that, I think, made it the most popular choice. The ease of design as well as making it, cloning it was extremely simple. And this also democratized the genetic manipulation. It gave everybody the opportunity. It was not now, talents were and zinc fingers were limited because zinc fingers, there were a lot of patents protecting them, and the design of the zinc fingers was not really known much outside because a lot of things were not published and they were in the hands of Sigma, the one of the major suppliers. And Thailand's also were working in certain groups very well, and certain groups could not set it up well. Whereas this one was everybody's tool, so everybody could use it. So I think that's the major impact that it has created. And what it has done is it has changed the pace of biological research with new possibilities to ask new questions. So, simple example I can give you is my whole PhD was generating one knockout. Now we could, some of my students have generated like 10, 15 knockouts, cell lines in their time. So it's pretty easy now. And the pace of biological research has changed, and the possibilities to ask new questions, because it's not just in the generating the knockouts. You can manipulate the genome the way you want. You can engineer it using the other associated genome engineering tools. So this has opened up possibilities which were not there before. And the commercial implications are also immense and are pretty visible in case of particularly in the crop science. In fact, the major application I feel is happening in the crop sciences, where people in the agricultural research have utilized it to generate large number of edited crop plants. Of course, many of them are, I think, under the consideration for testing and the release. But this is one area where it has picked up extensively. And also coming in the animal science, also in the farm animals, there have been a lot of CRISPR mutant animals that have been created, which are resistant to many viral diseases, like the African swine flu, swine fever, and many other diseases. Okay, and in the recent times, many of the gene therapy for monogenetic disorders, like the best example is sickle cell anemia, are in clinical trials, and many more are being added. Most of the monogenetic disorders are coming under this, and people are trying to treat. Particularly, the focus is on the hematopoietic lineage, because in case of hematopoietic lineage, the stem cells can be taken out, manipulated, and put back. That's one thing. Of course, that's the limit to use it in humans, not to go into the germline. So the germline is something not allowed in any country, and that's unethical. But at the somatic cells, this has been used in multiple places and for multiple disorders, and it's some of them are in the early stages of the clinical trials. So this brings to the end of my talk, and I would like to acknowledge that all the images are sourced from either Nobel website or from the internet, so nothing is of ours or original. Okay, and I would like to leave this with a serious caution that it's a very powerful technology that needs to be handled with responsibility. Because anything that's powerful, anything that's easily available, anything that can be easily handled, there is a good chance of misuse. So I think we all have to handle it with responsibility. So with this, I'll be happy to answer if there are any questions.
Yes, sir. I think let's bring on the questions now. So, Aditya asks that is there any method to predict any mutations which can happen while using CRISPR?
Yeah, so most of the, so as I said, off-target effects, these are what we call as. So there was a good paper in Nature Biotechnology where they predicted that what is the precision with which the CRISPR will lodge onto the locus you want and no other locus. Okay. And using this, they have designed the algorithms to design the guide RNAs. But in spite of it, there can be off targets. The only way you can know is there are two ways. One is go by a candidate approach. When you design the guides, you know that these are other 15 or 20 possible genes where my guide may lead it and cut the DNA. So you can specifically amplify those regions and sequence them. But given the cost of sequencing is low now, the best thing would be to go for a whole genome sequence of that cell line or the animal, so that you know that there is no off-target mutation that is created.
Okay, so we can sequence the whole genome in order to check
Yeah.
and predict the mutations.
Yeah. And in case of with genomes that are very well known, it's pretty straightforward. It's not difficult. Only in the genomes where the sequences are not available, the reference genome is not very well available, then there will be a trouble. If not, it's quite straightforward.
Oh, yes. Yes. And okay, by this, I think I'll move to the next question. I hope this was answered. Uh, Next question is by Sarah. She asks, is there any reverse CRISPR? Anything as such that instead of cutting out sequences from genome, it
Mhm.
adds sequences, or what would it be like? And or is there any CRISPR used for this same purpose?
So as I said, CRISPR's only, so the job of CRISPR's is, I actually like this an example to give you. It's like a letter with an address. If you want a letter to reach a specific person in Hyderabad, among all the people in the world, you would write the address with the country, place, road, name of the person, and the door number. That's exactly what the CRISPR does. If you take what the restriction enzymes, they will only write the country's name, so it can go into any city. That so this is the precision with which it gives you an address. The sequence will take you to the locus, and then it cuts there. That's it. CRISPRs do not do anything after that. After that, if you want a genetic manipulation, as I said, in non-homology and joining, it is a random thing. Then you will have to choose among multiple events. You have to choose the cell or the animal in which the event of your choice has happened. That is by sequencing that region. Or if you give a template or the donor DNA with a sequence of your choice with homology arms, it will go and stitch it in. This happens at a lower frequency, but it does happen. So you can insert bigger fragments or a gene. These are called knock-ins. You can do it using CRISPR. But there, the cut is only done by CRISPR, not that process of stitching it in. Process of stitching in is done by the cell itself, cell's repair system.
Oh, yeah, that's something new which we got to know. And okay, so CRISPR maybe it can be used for both purposes to
Yeah,
either
it is used for,
edit
yes,
and
for the
yeah,
purpose. Yeah,
yeah.
to
Okay,
delete or to knock in a sequence of your choice.
perfect. So moving on to the next question, Deepika asks that what is your take on the fruit about the patentship of the CRISPR technology?
Okay, the controversy seems to be slowing down, but it is still not done away. So if you see, the first paper was from Charpentier and Doudna, and they were also the first ones to file the patent. Okay, Feng Zhang came up with this, and they filed a patent later, but they paid a lot of charges to speed up. There is an option to speed up your patent, and that's why they were given the patent earlier. However, I think in the current scenario, the US patent is awarded to Feng Zhang and on priority, and the European one is awarded to Doudna and Charpentier. I think eventually it will be solved, but it doesn't look like has got any impact for the biologists. So it doesn't matter to us, but it I think will be of uh, implications to people who want to use it for commercial purpose.
Yeah, so for science and technology, maybe it's just a boon and we don't care who the patent goes to.
Yeah, but for applications, yes, it would matter in due course of time.
Okay. And sir, don't you think that it's a high time for the committee to have a Nobel Prize in biological sciences?
Medicine is biological science. There was nothing much distinguished. I don't think it's different.
What about no? So biological sciences would include plant sciences, microbial studies.
Yeah. Even those will get Nobel in under the medicine or in the chemistry. For example, this one is microbiology. It got
Yes,
under
sir.
chemistry. Of course, this is injustice to the chemists.
Yeah. Yes, sir. Okay. Moving on to the next question. Um, Ashutosh asks, I know that India's Feluda, if I'm pronouncing it right,
Mhm.
COVID-19 test works on CRISPR. But
Mhm.
instead of just diagnosing, can this technique also be employed in treatment against the coronavirus?
So the problem with treatments is you will, so how corona works, corona basically binds to ACE2, you will have to mutate the ACE2. That means you will have to mutate ACE2 in almost all your cells. That's not possible, isn't it?
Yes, sir.
So this one, although the tool works to manipulate the genome of mammalian cells, it's still not very efficient. It is of use only in case of monogenetic disorders where you can have some cells modified and that would take care of the body. Okay, so to it's not easy to treat viral diseases from using this. I don't think that's possible.
Mm-hmm. Okay. Moving on to the next question. A revolutionary technology like CRISPR can arise many ethical issues. So, how can a technology and ethics be balanced? This was asked by Bhavna.
Okay, so I think be it CRISPR or any other tools, ethical issues are of serious concern. It's like nuclear technology, you can use it for good or bad. I think it's dependent on how the society looks at it and how we as scientists look at it and build awareness that what are the dangers that are awaiting if we cross certain lines. Okay, in fact, if you listen to, in fact, I liked it. If you listen to the interview of the chairperson of the Nobel Committee, which awarded the one for chemistry, he brings back World War II because this is a tool that can be used to manipulate human genome. Now we have learnt our lessons from World War II, where where people were trying to create humans of a specific race or selective breeding and all these things. So those are the limits which we know are dangerous and we should never enter. We have historical lessons from them. I think not just this tool, any other tool which is so powerful should be handled carefully.
Yeah, I think an add on question to this would be that since you mentioned that CRISPR is used a lot to modify food crops, so
Yep.
how far do you think it's safe to modify our food crops, as in producing genetically modified crops? Because since there's no long term research or study, we can never be sure.
Yeah, I agree. So let's look at it like this. The green revolution and whatever the plants we eat these days are mostly hybrids. They are acceptable to us. You cross this with that and you are fine with it, isn't it?
Yeah,
And what is the selective breeding? Among the whole population, you may have some mutants that have arisen and you will try to selectively breed them and get them. Now that mutation could be one base missing or two base missing. If a CRISPR does it, you can't even tell that it was done by CRISPR. Okay, so
yeah.
it looks as natural or it is as natural as that. So why will we have difficulty accepting it? That's my point. Yes, if you have too much of genetic manipulation, you have put in some 2 3 genes or a large stretch of DNA from some other foreign organism, yes, then there should be stringent processes to see if the crop is safe or not. It's not that we outrightly ban it or stop it. There are processes that can be followed to see if that is safe or not. Otherwise, most of these manipulations, which are one or two bases, they are equivalent to the natural mutations that arise in any case. So it's very difficult to it's very difficult to distinguish it because there is no trace left behind how it happened. So I don't think that should be an issue. Only thing is, if you don't bring it into the regulation, there will be abuse of it. So it should be brought into regulation, but it should not be regulated heavily. It should be just because it's like a natural mutation. I don't think that should be strongly regulated, or there should be any hesitation to accept it.
Yeah, so I think like whatever modifications we try to make, if it's as close at, if it is as close as, or if we can mimic the natural modifications, it will not affect us in a bad way. But
Definitely,
if we, yeah, if we make like very, uh, I don't know, very big changes or just, uh, yeah, changes which are just not naturally possible, then it will definitely. cause
No,
harm.
then also you don't know if it will affect. You will have to take it through the process of seeing how safe the food is. If it is safe,
Yeah,
then we bother. If it's environmentally safe and safe for humans, safe for everyone, I don't think we should bother.
yeah, yeah, exactly. And I think in you
That
not
requires a lot of work,
yes.
basically.
And I think that the same mentality goes behind the anti-vaxxers group who are very popular in West. They just don't want to put any genetic modification to their gene, but they don't really understand that this is what our body does without any aid.
Corona infecting the cells itself is genetic material coming in from outside.
Exactly, yes, sir.
Yeah.
Yeah. Anyway, sir, let's not divert it to some other topic. Astha asks that since you mentioned that you have been using CRISPR in your labs, so have you had any trouble using CRISPR in your experiments? Can you share with us?
So honestly speaking, I didn't have much because, as you said, I am trained as a mouse genetist. We mouse genetists used to do this genetic manipulation very much before the CRISPRs. So ESLs have got a very high level of homologous recombination. So this was the only mammal where you could do the manipulation. So we were used to doing it. CRISPR only helps us to make it faster and quicker and scale up things because it further enhanced our homologous recombination capabilities. So we didn't really have much difficulty in adopting it and using it in our lab.
Okay. And I think this is one more question from the same person that uh, to what extent do you think CRISPR is successful in avoiding off-target effects?
So CRISPR has come a long way from the beginning till now. Based on the structural biology studies, people have come up with modified CRISPRs or the mutant versions of it, which have very low off-target effects. There is never you will be able to make it zero. You are only decreasing the possibilities by leaps and bounds. Now I think we have systems where these are almost minimal. And if it is a single base change, you don't even need to use a typical CRISPR. Instead, you use a base editor, the ones I mentioned in the end. You can, if you are changing one or two bases, you can use base editors which don't even cut the DNA.
Hmm.
So I think off-target is not anymore a serious concern,
Mm-hmm.
at least for the biologists and even for people who are involved in crop breeding and animal breeding, because even if there is an off-target mutation, you can breed it out in the subsequent generations.
Mm-hmm.
Only in therapy it might be of serious concern.
Yeah. And one more question that Pranita asked that any role of CRISPR in the production of synthetic genomes? That is it possible to construct a synthetic genome right from scratch using CRISPR?
I think yes, there have been some examples, and there have been also examples to use CRISPR as a sort of a hard disk. Okay, there was a beautiful paper which showed that using CRISPRs they could store the image of in the bacteria, image of a small image basically. So yes, they can be used for such things. So basically, it's a tool that can go into a specific DNA. So that specificity is the key of CRISPRs, going to a specific sequence. And later on, what you tag with Cas9, based on that, you will be able to use it for multiple purposes.
Okay, so maybe it is sort of similar to a question asked earlier that we can cut the DNA and add new nucleotides.
Yeah.
Same way we can, yeah, synthesize a genome from scratch.
Yeah.
Okay, so I think we can move to questions from faculty members in the GMeet. So please, I request all any faculty member who has a question to unmute themselves and ask.
Hi, Chandrasekhar Sharmistha here.
Misha Mista.
Hi. I just thought I'll ask for generally them. If you can, because there may be a mixed audience here, tell us a little bit about Feluda and how it works. Touching CRISPR and not touching Feluda is something.
Okay. So Feluda is based on a similar system that was published by Feng Zhang some time back. Um, so that was also named Sherlock. So basically,
Yes.
the way it works is either you want to detect in case of RNA or DNA, you can, for example, in case of Feluda, it's trying to detect the RNA. So it doesn't directly detect the RNA. Basically, you have primers which will be used for reverse transcription of the RNA from the virus, or it could be from any bacteria, let's say. Then you carry out a PCR to amplify the genome. Once the genome is, or the specific PCR product, let's say 100 or 200 bases, then you have the Cas protein and the guide RNA, which is specific to that. When you put them together in the mixture, so now the Cas with the guide RNA will be able to bind to the DNA, open it up, and in this case, it's mostly the dead Cas9, so it will stick to it. Now you can use any other detection systems like a paper-based one, or you can use any flow devices to detect the binding of this DNA to the Cas9 and the guide RNA. So this complex can be detected. So different tools have been used to detect. People use even fluorescence. Some use the strip test. I think Feluda is based on the flow device, lateral flow device. So then you will see the reaction happening, because you can have your DNA tagged to an HRP or a streptavidin, which later on can pick up an HRP from streptavidin HRP, and you will see the reaction happening when it binds to the Cas9. So that's how it works.
Thank you so much. And I hope that is helpful for many. Thank you so much. I really enjoyed listening to you.
You're welcome.
Yes, sir, it was a wonderful session. I thoroughly enjoyed it personally.
Thank
And
you.
thank you so much, sir. Sir, so I would like to ask again if there are any questions from the faculty members in the meet, or maybe we can end it here.
Ex- Yeah, there was this question about any idea on using two caste systems to generate large deletions.
You don't need two Cas systems. In fact, as I said, the Cas can only cut the DNA. It depends on where you put your guides. In fact, 100 kb, 200 kb can be deleted easily. 100 kbs have been deleted in our lab and many other labs. So it's just where you place the. If you place them two guides very far away, the distance will be cut. Everything in between is gone.
Okay. I
Okay.
hope that is done. I think you can conclude.
Yes, sir. Thank you so much, sir, for your valuable time. I know that it was sort of difficult for you to make time for us, but we are very thankful for us to joining us and giving us a detailed insight on CRISPR. Uh,
Thank you very much. It was a pleasure talking to you guys.
Thank you so much, sir. And we'll be having two more talks in the Nobelium webinar series, which will be the first talk for the day for tomorrow will be on the discovery of hepatitis C virus, and the second talk will be the economics Nobel Prize talk, which will be on the improvements of the auction theory. I request all of you, if you have time, please do join us and please do put in your valuable questions. Thank you so much, everyone, for being a part for the day. You can follow the Junior Science Club on Instagram on the handle at the rate GSC underscore UOH. Meet all of you tomorrow. Thank you.